Welcome to Sleepy Science Odyssey. Tonight, nature, the living systems that cover Earth's surface, from microscopic fungi threading through soil to towering forests that shape continental climates, from the symbiotic partnerships hidden underground to the cycles of nutrients that sustain all life. We'll move slowly through ecosystems and biomes, exploring how plants produce the oxygen you breathe, how forests create their own weather patterns, how organisms interact in webs of astonishing complexity, and how natural processes maintain the delicate balance that makes Earth habitable. From the smallest bacteria fixing nitrogen in soil to vast boreal forests storing carbon across continents, from desert organisms surviving on millimeters of annual rainfall to rainforests where a single tree hosts thousands of species. If you're new here, this channel is for people who need something to think about before they can stop thinking. Real natural science, no exaggeration, just quiet facts in the dark. Let's begin. Plants perform photosynthesis, converting light energy into chemical energy stored in glucose. Green plants, algae, and some bacteria capture sunlight and transform it into chemical bonds that power nearly all life on Earth. This process, photosynthesis, occurs in chloroplasts, specialized organelles containing chlorophyll that gives plants their green color. Chlorophyll absorbs red and blue light while reflecting green wavelengths, explaining why most plants appear green to our eyes. But there's more complexity than this simple explanation suggests. Chlorophyll actually exists in several forms, with chlorophyll A and chlorophyll B being the most common in plants, each absorbing slightly different wavelengths and working together to capture a broader spectrum of light energy. The molecular structure of chlorophyll resembles hemoglobin in blood, both built around a ring structure called a porphyrin, though chlorophyll's central atom is magnesium while hemoglobin's is iron. The process occurs in two stages within the chloroplast. A double-membraned organelle descended from ancient cyanobacteria that were engulfed by early eukaryotic cells over 1 billion years ago in an endosymbiotic event that fundamentally changed the trajectory of life on Earth. Light-dependent reactions capture energy from photons, splitting water molecules to release oxygen and generating energy-carrying molecules ATP and NADPH. These reactions occur in the thylakoid membranes, stacked structures within chloroplasts that create enormous surface area for photosynthetic machinery. A single chloroplast can contain 40-60 thylakoids stacked like pancakes, and a single leaf cell might contain 5,100 chloroplasts, creating truly impressive total membrane surface area dedicated to capturing light. Light-independent reactions, the Calvin cycle, use this stored energy to fix carbon dioxide into organic molecules, occurring in the stroma, the fluid-filled space surrounding thylakoids. The overall equation appears simple. Carbon dioxide plus water plus light yields glucose plus oxygen. But the molecular machinery involves hundreds of proteins working in coordinated sequences, with feedback mechanisms regulating the process in response to light intensity, temperature, carbon dioxide availability, and the plant's internal needs. The efficiency of photosynthesis is remarkable, yet limited by fundamental physical and biochemical constraints. Only about 1–3% of incident solar energy is converted into chemical energy in most plants under optimal field conditions, with theoretical maximum efficiency around 4–6% for C3 plants and slightly higher for C4 plants. This might seem inefficient compared to photovoltaic solar panels that can achieve 15–20% efficiency or more. but it represents billions of years of evolutionary optimization under multiple constraints, including the need to dissipate excess light energy that could damage cellular machinery through the generation of reactive oxygen species. The thermodynamic limits of chemical reactions that cannot violate the laws of physics, and the requirement to balance carbon gain against water loss through stomata. Different photosynthetic pathways, C3, C4, and CAM, represent different evolutionary solutions to these constraints, with each optimized for different environmental conditions, and each involving different trade-offs between efficiency, water use, and energetic costs. The light-dependent reactions are truly fascinating when examined in detail. When a photon strikes a chlorophyll molecule in the antenna complex, a cluster of hundreds of pigment molecules surrounding the reaction center, it excites an electron to a higher energy state. This excitation energy is passed between pigment molecules like a hot potato until it reaches the reaction center, a special pair of chlorophyll molecules where the actual chemistry occurs. The excited electron is captured by an electron acceptor, beginning a cascade down an electron transport chain, a series of protein complexes embedded in the thylakoid membrane. As electrons flow down this chain, they release energy that pumps hydrogen ions across the membrane, creating a gradient. This gradient drives ATP synthase, a molecular turbine that rotates as hydrogen ions flow through it, synthesizing ATP from ADP and phosphate. Meanwhile, the electron eventually reaches photosystem I, where it's re-energized by another photon and used to reduce NADP+ to NADPH. The electrons lost from the reaction center are replaced by splitting water molecules, releasing oxygen as a byproduct. Photosynthesis sustains Earth's oxygen atmosphere, maintaining the 21% oxygen concentration we depend on. Before photosynthetic organisms evolved about 3 billion years ago, Earth's atmosphere contained virtually no free oxygen. The early atmosphere likely consisted primarily of nitrogen, carbon dioxide, water vapor, and trace amounts of other gases, with methane possibly playing an important role as a greenhouse gas, keeping early Earth warm despite the sun being about 30% dimmer than today, a puzzle known as the faint young sun paradox. Cyanobacteria, among Earth's earliest organisms and still abundant today in oceans and freshwater, began producing oxygen as a waste product of their metabolism through oxygenic photosynthesis, a revolutionary innovation that changed everything. This oxygen initially reacted with dissolved iron in oceans, precipitating as iron oxide deposits visible today as banded iron formations, distinctive layered rocks with alternating iron-rich and iron-poor bands that record the gradual oxygenation of Earth's oceans over hundreds of millions of years. These banded iron formations, found on every continent and mined extensively for iron ore, tell a story of planetary transformation. For perhaps 1 billion years, oxygen produced by photosynthesis was consumed by reacting with reduced chemicals in the environment, iron in seawater, sulfur compounds, and organic matter. During this period, oxygen concentrations remained negligibly low, but the groundwork was being laid for the dramatic changes to come. Eventually, around 2.4 billion years ago, in an event called the Great Oxidation Event, or the oxygen catastrophe, oxygen began accumulating in the atmosphere after reacting with all readily available reduced chemicals in oceans and rocks. This fundamentally transformed Earth in ways we're still uncovering. It likely triggered what may have been the first global glaciation, the Huronian glaciation, as methane, a potent greenhouse gas, was oxidized to carbon dioxide, a weaker greenhouse gas, possibly creating a snowball Earth scenario where ice covered much of the planet for millions of years. More importantly for the eventual evolution of complex life, the accumulation of oxygen enabled aerobic life forms, organisms that use oxygen for metabolism in the process called cellular respiration. Aerobic respiration extracts far more energy from organic molecules than anaerobic processes, approximately 18 times more ATP molecules per glucose molecule compared to anaerobic fermentation. This dramatic increase in available energy provided the foundation for the evolution of complex, multicellular life. Single-celled organisms can survive on the meager energy from fermentation, but large, complex organisms with specialized tissues and high-energy demanding processes like nervous systems and muscular contraction require the abundant energy that only aerobic respiration can provide. Without the oxygen produced by photosynthesis accumulating over billions of years, animals, including humans, could not exist. Every breath you take contains oxygen that some photosynthetic organism produced, possibly recently from a nearby tree or crop field, possibly years ago from a marine phytoplankton cell in the Pacific Ocean, circulated globally through atmospheric mixing. Today, photosynthesis produces approximately 130 billion tons of organic material annually on land through net primary production. The amount of carbon fixed by plants minus what they consume through their own respiration for growth, maintenance, and reproduction. Marine phytoplankton contributes similar amounts, approximately 130 billion tons, making the oceans roughly as productive as land in total despite covering 71% of Earth's surface. Though productivity is concentrated in coastal zones, upwelling regions, and certain oceanic areas rather than being evenly distributed. The open ocean, despite its vast area, is relatively unproductive per unit area, often called a biological desert due to nutrient limitation, though even low productivity across enormous area adds up to substantial total production. This global productivity forms the base of food webs, with herbivores consuming plants and gaining access to the chemical energy stored in plant tissues through their own digestive processes. Carnivores consuming herbivores and accessing energy passed up the food chain with substantial losses at each trophic level due to the second law of thermodynamics, and decomposers breaking down dead organisms at all levels to recycle nutrients back into forms that plants can use to continue the cycle. Without photosynthesis continuously capturing energy from sunlight and converting it into chemical forms, this entire edifice of life would collapse within years as stored energy was exhausted through respiration and decomposition. The standing biomass of Earth, all the living organic matter in plants, animals, and microbes, represents accumulated photosynthetic production over years to centuries. If photosynthesis stopped, this biomass would be consumed and decomposed relatively quickly on geological timescales, with most disappearing within decades as organisms starved and decomposers processed organic matter. The interconnectedness of life through energy and nutrient flows means that virtually all organisms ultimately depend on photosynthesis, with only a few exceptions like chemosynthetic communities at deep-sea hydrothermal vents that obtain energy from chemical reactions instead of sunlight. Forests cover about 30% of Earth's land area and contain approximately 80% of terrestrial biodiversity. Forests exist wherever temperature and precipitation allow tree growth, creating environments where woody plants can dominate over grasses and shrubs. They range from tropical rainforests near the equator, receiving over 2,000 millimeters of rain annually with temperatures consistently between 20-28 degrees C, to boreal forests near Arctic regions, where trees must survive 6-month winters with temperatures dropping to degrees C or colder and growing seasons compressed into 3-4 months of the year. These diverse ecosystems provide habitat for countless species while regulating climate through multiple mechanisms, producing oxygen that maintains atmospheric composition, storing carbon in both living biomass and accumulated dead organic matter in soil, cycling nutrients between soil and atmosphere through uptake and return, and protecting watersheds by intercepting rainfall before it strikes the ground, reducing erosion through root networks that hold soil, and regulating stream flow by absorbing water during storms and releasing it gradually. The global extent of forests has declined substantially from pre-agricultural times. Estimates suggest roughly half of the forests that existed 8,000 years ago, before widespread human agriculture transformed landscapes, have been cleared primarily for agriculture, with deforestation continuing today at approximately 10 million hectares annually, mostly in tropical regions where expanding agriculture for soybeans, palm oil, cattle ranching, and smallholder farming, along with logging and development, continues converting forests to other land uses despite growing recognition of forests' value. The loss of forests represents more than just lost trees. It's the loss of entire ecosystems with thousands of species, many found nowhere else, many not yet described by science, and some potentially going extinct before we even know they existed. Recent research suggests we're losing species faster than we're discovering them, particularly in tropical regions where diversity is highest, but also where deforestation rates are fastest. Each species lost represents millions of years of evolutionary history erased, unique genetic information destroyed, and potential contributions to ecosystem function and human benefit eliminated. Some of these species might have provided medicines. Many current drugs derive from plants and other organisms, and many more potential medicines remain undiscovered. Some might have provided insights into solving biological or engineering problems. Biomimicry has given us Velcro from burrs, improved aerodynamics from kingfisher beaks, and adhesives from gecko feet. Some might have been critical for ecosystem function in ways we don't yet understand because we're still learning how complex ecosystems maintain themselves through thousands of interacting species and processes. Tropical rainforests occur near the equator where rainfall exceeds 2,000 millimeters annually and temperatures remain warm year-round, typically between 20-28 degrees C with minimal seasonal variation. The daily temperature range often exceeds the annual temperature range. Despite covering only 6-7% of Earth's land surface, currently about 9 million square kilometers concentrated in the Amazon, Congo, and Southeast Asian basins, they contain over half of all plant and animal species estimated to exist on Earth. This makes them the most biodiverse terrestrial ecosystems on the planet, rivaling or exceeding even coral reefs in species richness per unit area. A single hectare of Amazon rainforest might contain 300 tree species, more than all native tree species in North America, north of Mexico, which numbers around 700 species across the entire continent. This extraordinary diversity results from multiple interacting factors that have operated over millions of years. Stable climates over long timescales allow specialization. Without the extinction, events that periodic glaciations caused in temperate regions. Tropical regions remained warm and wet even during ice ages, providing refuges where species survived and diversified. High productivity from year-round growing conditions provides abundant energy to support numerous species without the energetic constraints that limit diversity in harsher climates. Complex three-dimensional structure creates countless ecological niches. Different species specialize on different canopy layers, different light conditions, different substrates, different food resources, creating fine-scale partitioning that allows many species to coexist. And intense species interactions including predation, herbivory, competition, and mutualisms promote specialization and diversification. Arms races between plants and herbivores drive chemical defense evolution and herbivore counteradaptations. Pollination mutualisms drive flower and pollinator coevolution, and competition drives niche differentiation. Rainforest structure includes multiple distinct layers, each with characteristic microclimatic conditions and species communities that differ dramatically over vertical distances of just tens of meters. The emergent layer features scattered tall trees reaching 50-60 meters or more, occasionally exceeding 80 meters in some forests, their crowns poking above the main canopy like isolated giants fully exposed to full sunlight that can be intense enough to damage leaves, high temperatures that can exceed 35 degrees C in direct sun, and strong winds that can reach speeds rarely experienced by understory plants. These emergent trees often have distinctive adaptations including buttress roots, large, thin, stabilizing structures extending from the trunk base that help support tall trees in relatively shallow soil, where deep taproots are prevented by impermeable clay layers or high water tables. Some buttresses extend 5–10 meters up the trunk and out from the base, creating dramatic architectural forms. The tallest tropical trees exceed 100 meters in some Asian dipterocarp forests, rivaling temperate conifers in height despite the physical challenges of lifting water and supporting tall structures in less dense tropical woods. The canopy layer at 30–45 meters forms a continuous or nearly continuous roof where most photosynthesis occurs, and where the majority of rainforest animals live, feeding on abundant fruits, flowers, leaves, nectar, and each other in a three-dimensional world rarely touching the ground. Canopy trees' crowns interlock or nearly touch, creating pathways for arboreal animals like monkeys, sloths, tree kangaroos, squirrels, and countless birds to move through the forest traveling hundreds of meters without descending to the ground where predators wait. The canopy microclimate differs dramatically from the forest floor. Higher temperatures, lower humidity, more sunlight, stronger winds, greater temperature fluctuations. Species living in the canopy are adapted to these conditions and often cannot survive at ground level where conditions are too dark and humid. The canopy hosts enormous biodiversity, including not just animals, but also epiphytes, plants growing on other plants without parasitizing them, including orchids, bromeliads, ferns, mosses, and even small trees that germinate in accumulated organic matter on branches. A single large tree might support hundreds of epiphyte species, creating aerial gardens with their own associated insects, spiders, frogs, and microorganisms. Below the canopy lies the understory, receiving perhaps 2–5% of sunlight that reaches the canopy top after being filtered through multiple layers of leaves above. This dim environment, with light levels sometimes as low as 0.5% of full sunlight, supports shade-tolerant species, small trees, shrubs, and saplings of canopy species waiting decades or even a century for gaps to open overhead, allowing rapid growth into the canopy when opportunity arrives. These understory trees don't just sit dormant, they grow slowly, producing leaves, flowers, and fruits, but they cannot achieve reproductive maturity and produce abundant seeds until they reach the canopy and access full light. When a canopy tree falls, creating a gap, understory trees race to fill it through rapid growth, with the fastest growers capturing the space and suppressing slower neighbors. This gap phase regeneration creates a mosaic of different-aged patches across the forest, contributing to overall diversity. Understory plants often have large, dark green leaves, maximizing light capture in low-light conditions. Large surface area captures more of the limited photons available, and dark color from high chlorophyll content absorbs light efficiently. Some understory plants have iridescent blue leaves from specialized structures that reflect blue light to photosynthetic layers below, increasing light capture efficiency. The shrub layer near the forest floor hosts specialized species adapted to extreme shade, including plants with dramatic leaf shapes and sizes, some with leaves exceeding a meter in length to maximize light interception. The forest floor itself is relatively open compared to temperate forests, without the dense understory of shrubs characteristic of many temperate deciduous forests, because insufficient light reaches the ground to support dense growth. However, the floor is covered with decomposing organic matter, leaves, branches, fruits, flowers, animal remains, and feces, rapidly broken down by fungi, bacteria, and invertebrates in the warm, humid conditions. Decomposition is so rapid that organic matter often disappears within weeks. A leaf falling from the canopy might be completely decomposed, and its nutrients released and absorbed by plants within a month. This rapid cycling prevents accumulation of thick organic layers like those in temperate or boreal forests, where cold or dry conditions slow decomposition. Walk through a temperate forest, and you'll find thick leaf litter accumulated over years. Walk through a tropical rainforest, and you'll find only a thin layer of recent fallen material actively being decomposed. Paradoxically, despite lush vegetation creating the impression of fertility, tropical rainforest soils are often nutrient-poor, heavily weathered over millions of years of intensive chemical weathering, and acidic from organic acids produced by roots and decomposers. The high rainfall that supports forests also leaches nutrients from soil, carrying them downward in percolating water beyond root reach or washing them laterally into streams. Nutrients including nitrogen, phosphorus, potassium, calcium, and magnesium are continuously lost from the system through leaching, though this is partially offset by inputs from rock weathering and atmospheric deposition. The forest's high productivity depends on tight, efficient nutrient cycling rather than rich soil reserves. Nutrients exist primarily in living biomass rather than soil, opposite the pattern in temperate and boreal forests where substantial nutrients are stored in organic matter and mineral soil. When a leaf falls, its nutrients are quickly released through decomposition and immediately absorbed by the dense root mat near the surface, preventing them from leaching away. This recycling is so efficient that rainforests can maintain productivity on soils that would be considered infertile for agriculture. When rainforests are cleared for agriculture, this illusion of fertility is shattered. Without continuous recycling, the limited soil nutrients are quickly depleted, typically within 2-3 years for traditional slash-and-burn agriculture, or 5-7 years for more intensive systems with fertilizer inputs. Yields decline, farmers abandon fields and clear new forest, beginning the cycle again. The cleared land doesn't simply revert to forest. Erosion removes topsoil, compaction from machinery or cattle degrades soil structure, and invasion by aggressive grasses or shrubs prevents tree regeneration. After a few cycles of clearing and abandonment, land can become permanently degraded, supporting neither forests nor sustained agriculture without massive inputs of fertilizers, lime to correct acidity, and tillage to break compacted layers. This explains why much former rainforest becomes degraded land, landscapes of scrubby second growth, pastures of poor quality, or even near bare ground in extreme cases, representing permanent loss of both forest ecosystems and productive agricultural potential. The tragedy is that this degradation is unnecessary. Agroforestry systems that integrate crops with trees can maintain productivity for decades or indefinitely by mimicking natural forest nutrient cycling while still producing food, demonstrating that tropical regions can support human needs without destroying forests if we design systems that work with ecological processes rather than against them. Temperate forests occur in mid-latitudes where seasons are distinct, with warm summers allowing active growth and cold winters requiring dormancy or adaptations to survive freezing temperatures. Deciduous forests, dominant in eastern North America from the Great Lakes to the Gulf of Mexico, in Europe from the Atlantic to the Ural Mountains, and in Eastern Asia including China, Korea, and Japan, drop leaves in autumn as an adaptation to winter cold and limited water availability when soil water freezes. Before shedding, trees reabsorb valuable nutrients from leaves, particularly nitrogen, phosphorus, and potassium that are mobile within plants and can be translocated through the phloem, the vascular tissue that transports sugars and other compounds, moving them to roots and woody tissues for storage over winter. This nutrient resorption is highly efficient, with trees recovering 50–90% of nitrogen and 50–70% of phosphorus from senescing leaves, representing an important adaptation that allows trees to invest in new leaves each spring without requiring equivalent nutrient uptake from soil. The efficiency of resorption varies among species, and with nutrient availability, trees on infertile soils tend to have higher resorption efficiency because nutrients are precious and worth recovering, while trees on fertile soils are less efficient because nutrients are readily available from soil. The falling leaves create a thick layer on the forest floor that decomposes more slowly than in tropical forests. A leaf might persist for a year or more before completely decomposing, with tough, lignin-rich leaves from oak taking longer than thin, nitrogen-rich leaves from ash or maple. This slower decomposition builds rich topsoil over centuries and millennia, creating the thick, dark, a horizon's characteristic of deciduous forest soils. These temperate deciduous forest soils classified as alphasols and malasols in the soil taxonomy system, are among the most fertile on Earth, which is why much of the original deciduous forest in Europe, eastern North America, and eastern China has been converted to agriculture. The famous corn belt of the American Midwest developed on malasols formed under prairie and oak savanna, not continuous forest, but adjacent regions in Ohio, Indiana, and Illinois had dense deciduous forest on alphasols that are also highly productive. European agriculture has exploited deciduous forest soils for millennia, with clearing beginning in Neolithic times and accelerating through Roman times and the medieval period. By the time of the Industrial Revolution, much of Western and Central Europe had already been cleared, with forests reduced to fragments. The seasonal temperature variation in temperate regions creates dramatic annual cycles that structure biological communities in ways unknown in tropical regions. Spring brings a flush of wildflowers, spring ephemerals like trilliums, bloodroot, Virginia bluebells, wild ginger, hepatica, and Dutchman's breeches, that complete most of their annual growth, photosynthesis, and reproduction in the few weeks between snowmelt and canopy closure. These species must complete their life cycle in the brief window when sunlight reaches the forest floor, before the tree canopy leafs out and shades them for the rest of the growing season. Their adaptations are remarkable and include bulbs or rhizomes storing energy underground accumulated from previous years, allowing rapid growth when conditions permit without requiring time to build up resources from scratch. They exhibit rapid growth enabled by relatively warm soil temperatures in spring after winter cold has passed, emerging quickly, expanding leaves rapidly, sometimes growing several centimeters per day. And their flowers appear before or with leaves, prioritizing reproduction in the limited time available rather than first building photosynthetic machinery to support reproduction later. These spring ephemerals provide crucial early-season resources for pollinators emerging from dormancy or hibernation, with some specialized relationships between particular plant and pollinator species that have co-evolved over millions of years. Summer in deciduous forests is characterized by the full, dense canopy blocking most light from reaching the understory and ground, creating the dim, cool conditions characteristic of these forests in popular imagination. Understory trees and shrubs adapted to shade grow slowly, producing leaves and flowers when conditions permit, while herbaceous plants either persist in deep shade with very low growth rates or go dormant after completing spring growth. Productivity is high during summer as trees photosynthesize under optimal conditions: long days, warm temperatures, abundant soil moisture from spring rains. Animals are active, taking advantage of abundant food resources to grow and reproduce. Birds nest and raise young, timing reproduction to coincide with peak caterpillar abundance when tree leaves are young and tender. Mammals raise young born in spring, feeding them through summer on abundant vegetation, insects, or prey. Insects complete multiple generations, with populations building to peak levels by late summer. Autumn transforms deciduous forests into spectacular displays of color as leaves senesce before falling. The colors aren't simply beautiful, they're symptoms of underlying biochemical processes preparing trees for winter. Chlorophyll, the green pigment dominating spring and summer leaves, breaks down in autumn, revealing other pigments that were present all along but masked by chlorophyll's intense color. Yellow and orange colors from carotenoids, red and purple colors from newly synthesized anthocyanins produced in response to bright light, and cool temperatures acting on sugars trapped in leaves, as nutrient resorption reduces the capacity to transport them. The adaptive function of autumn colors, particularly reds, is debated. They may protect remaining chlorophyll from light damage during the resorption period, or they may signal to aphids that these leaves are well defended and unpalatable, deterring egg-laying that would harm the tree next spring. Whatever their function, autumn colors provide one of nature's great spectacles, drawing millions of leaf peepers to New England, Appalachia, the Great Lakes region, and other deciduous forest areas to witness the transformation. Winter strips deciduous forests to their structural bones, bare branches silhouetted against gray skies, the complex three-dimensional architecture visible without the masking layer of leaves. Snow may blanket the ground, insulating soil and roots from the most extreme cold. Trees are dormant, respiration reduced to minimal levels, no growth occurring but not dead. Cells remain alive, preventing freezing through supercooling where water remains liquid below its normal freezing point, or accumulating sugars and other compounds that lower the freezing point, or tolerating ice formation in extracellular spaces while protecting cells themselves from ice crystals that would rupture membranes. Certain animals also remain active through winter. Some birds remain year-round, switching from insect diets in summer to seed and berry diets in winter. Some mammals remain active, relying on thick fur, subcutaneous fat, and behavioral adaptations like nesting in insulated sites. And many invertebrates overwinter as eggs or pupae, entering diapause, a programmed dormancy triggered by day-length changes in autumn that prepares them for winter months before cold arrives. Temperate coniferous forests, dominated by pines, firs, spruces, and hemlocks rather than deciduous broadleaf trees, occur in regions with particular conditions. Cooler temperatures where conifers have competitive advantages. Less fertile soils, particularly sandy or acidic soils where conifers tolerate infertility better than most broadleaf trees. Or disturbance regimes like frequent fires, where conifers' thick bark and serotinous cones that open in fire heat allow them to persist and regenerate. The Pacific Northwest temperate rainforests of North America, extending from northern California through Oregon, Washington, British Columbia, and into southeast Alaska, dominated by Douglas fir, western hemlock, Sitka spruce, and western red cedar, are among the most productive forests on Earth, accumulating biomass exceeding even tropical rainforests in some locations. These forests receive abundant rainfall, often 2,300 millimeters annually in lowland areas, exceeding 5,000 millimeters in some mountain valleys that funnel moisture-laden air from the Pacific, and mild temperatures year-round due to maritime influence from the Pacific Ocean, with winter lows rarely below degrees C and summer highs rarely above 25 degrees C, creating ideal growing conditions for the giant conifers. Trees in these forests can reach enormous sizes unmatched by temperate deciduous trees or even most tropical trees. Douglas firs routinely exceed 60 meters tall and can reach over 100 meters, with the tallest recorded at 99.4 meters before the top broke off, possibly reaching 121.30 meters before damage. Western red cedars grow slower but live longer, commonly exceeding 500 years and occasionally exceeding 1,000 years, with some trees dated to over 1,400 years, alive since before the fall of Rome. Sitka spruces become the largest spruces in the world, with the largest specimens exceeding 95 meters tall and 5 meters in diameter, trees of truly awe-inspiring dimensions that must be experienced to fully appreciate. The mild, wet climate supports abundant mosses and ferns that carpet the forest floor in thick, lush mats and festoon branches with hanging gardens, creating a green, humid world even in the dim light beneath the dense canopy. Some ancient trees support entire ecosystems on their branches. Accumulations of fallen needles, branches, and organic matter over centuries create soil layers meters thick in some large branch crotches, supporting herbs, shrubs, and even small trees growing 50 meters above the ground in what are essentially aerial islands of habitat disconnected from the ground. These ancient forests, sometimes called old-growth forests, though that term encompasses many forest types, develop complex structures over centuries that differ fundamentally from young forests. Multiple canopy layers create three-dimensional habitat complexity. Large dead trees, both standing snags and fallen logs, provide crucial wildlife habitat. Cavities in snags for nesting and denning, logs as nurse logs where tree seedlings establish in elevated microsites protected from competition and browsing. Gaps created by tree falls allow light penetration, creating small patches of different aged regeneration. And the accumulation of organic matter creates deep, spongy soils that absorb and hold water. These structural features take centuries to develop and cannot be recreated through planting. Young plantations lack snags, logs, canopy complexity, and developed soils that characterize old forests. The recognition that old-growth forests have unique ecological values beyond timber production has driven conservation efforts, though intense conflict continues between those prioritizing timber harvest and those prioritizing conservation. Boreal forests, also called taiga, form a vast belt across northern North America from Alaska through Canada, across Scandinavia, and through Russia from the Baltic to the Pacific, representing the largest terrestrial biome and covering approximately 17% of Earth's land surface, roughly 15 million square kilometers, an area larger than the United States. These forests are dominated by coniferous trees, spruces including white, black, and Norway spruce, firs including balsam fir and Siberian fir, pines including jack pine, lodgepole pine, and Scots pine, and larches including tamarack and Dahurian larch, the latter unusually deciduous among conifers, adapted to short growing seasons of only 3-4 months in many areas, cold winters reaching -40 degrees C to -50 degrees C, or even colder in parts of Siberia, where the coldest inhabited places on Earth experience winters below degrees C and acidic. Nutrient-poor soils that develop slowly in the cold climate, where weathering and decomposition proceed at glacial pace. Coniferous needles are evergreen in most species, allowing photosynthesis to begin immediately when conditions permit in spring, without the time and energy cost of growing an entire new canopy of leaves. A crucial advantage when the growing season is so short that dedicating weeks to leaf production would substantially reduce the period available for photosynthesis and growth. Some species like larches are deciduous, dropping needles in autumn as an adaptation to the harshest climates where even evergreen needles would suffer too much winter damage, making replacement cost-effective. Needles' small surface area and thick, waxy cuticle reduce water loss, important because frozen soil makes water unavailable in winter despite high water content. The water is locked in ice crystals, as unavailable to plants as if they were in a desert. This physiological drought means that evergreen needles still exposed to dry winter air and occasional warm, Sunny days that would drive transpiration must minimize water loss to prevent desiccation. Needles also contain compounds that protect them from freezing damage, lower the temperature at which ice forms, and prevent ice crystals that do form from rupturing cells. The boreal forest's dark green canopy absorbs most incoming sunlight, typically 90–95%, compared to lighter-colored vegetation or bare ground that might reflect 30–50%, warming the region and affecting global climate in ways that are only beginning to be fully understood through improved climate models and observations. This low albedo compared to tundra or snow-covered ground means boreal forests absorb more solar radiation than alternative land covers, which warms the surface locally, but also contributes to regional and global warming through heat transfer to the atmosphere. This warming effect is particularly pronounced in spring, when snow remains on ground but dark conifer canopies emerging from snow absorb sunlight and warm the air more rapidly than would occur with snow-covered tundra. This albedo effect partially offsets the cooling effect of carbon storage, leading to complex debates about the net climatic impact of boreal forests and whether policies should encourage boreal forest expansion as carbon sinks or recognize that albedo effects might make boreal forests less beneficial for climate than forests at lower latitudes. These forests store enormous amounts of carbon in multiple pools: living tree biomass, dead standing and fallen wood, roots, forest floor organic matter, and soil organic matter extending to considerable depth. Cold temperatures slow decomposition dramatically, allowing organic matter to accumulate despite relatively low productivity compared to tropical or temperate forests. Annual net primary production in boreal forests is typically 200–400 grams of carbon per square meter per year, compared to 1,001,500 in tropical forests, or 600,100 in temperate forests. But because decomposition is even slower, organic matter accumulates. Some boreal forests have organic layers, the O horizon combining fresh litter and partially decomposed material, exceeding 1 meter thick, accumulated over thousands of years since glacial retreat exposed the landscape roughly 10,000 years ago. Peatlands scattered throughout the boreal zone have accumulated organic layers 10 meters thick in some locations, representing massive carbon storage that would release enormous quantities of CO2 and methane if warming and drying cause decomposition to accelerate or fires to penetrate into the peat, both of which are occurring with increasing frequency as climate changes. The balance between carbon accumulation through productivity and carbon loss through decomposition and fire is sensitive to temperature, making boreal forests and peatlands potential climate tipping points, where warming could trigger positive feedbacks that release stored carbon and accelerate warming further. Already, recent decades have seen increased fire frequency and severity in some boreal regions, with fires burning larger areas and occasionally penetrating deep into peat, releasing carbon accumulated over millennia within weeks. Thawing permafrost, permanently frozen ground underlying much of the boreal zone, is also accelerating, exposing previously frozen organic matter to decomposition and potentially destabilizing forests as ground subsides unevenly, creating drunken forests with tilted trees and disrupted drainage. These changes could transform boreal regions from carbon sinks, which they've been for thousands of years, as forests and peatlands accumulated carbon faster than fires and decomposition released it into carbon sources that accelerate climate change, creating feedback loops that are challenging to reverse once initiated. A single large tree can transpire hundreds of liters of water daily, moving water from soil to atmosphere. Trees are sophisticated hydraulic systems, moving water from soil to atmosphere through their tissues in a continuous flow driven primarily by evaporation from leaves. This might seem like a simple physical process. Water evaporates, creating a deficit that pulls more water from below. But the mechanisms operating inside trees are far more complex and remarkable than this simple description suggests. Transpiration, the evaporation of water from leaf surfaces through stomata. Microscopic pores, typically 1040 micrometers long, that can open and close in response to environmental conditions, internal plant signals, and circadian rhythms, creates negative pressure or tension in the water column within xylem vessels, the specialized water-conducting tissues that run from finest root tips through trunk and branches to the smallest leaf veins. As water evaporates from the moist cell walls of mesophyll cells inside leaves, the spongy photosynthetic tissue between upper and lower leaf surfaces, and diffuses out through open stomata into the surrounding air, creating a deficit that pulls water from the xylem in leaf veins. This tension is transmitted down the continuous water column in xylem vessels, which are essentially hollow tubes formed from dead cells whose end walls have dissolved during development, creating continuous conduits that can extend from roots to leaves without interruption, pulling water upward from roots through trunk and branches to leaves against both the weight of the water column itself and frictional resistance from vessel walls and any constrictions or imperfections in the conducting system. The mechanism seems almost impossible when you consider the physics involved. Lifting water tens of meters in medium-sized trees, or over 100 meters in the tallest trees using only evaporation, without any pumps, without any mechanical assistance, without any energy input except the heat energy driving evaporation itself. The tallest tree currently living is Hyperion, a coast redwood in California measured at 115.92 meters in 2019, and fossil evidence suggests even taller trees existed in the past. The tallest recorded tree was a mountain ash in Australia measured at 132.6 meters in 1872 before it fell. Moving water to the tops of such trees requires overcoming approximately 1.113 megapascals of pressure from gravity alone, about 1113 times atmospheric pressure, plus additional pressure to overcome resistance to flow through xylem vessels. Yet trees accomplish this routinely through exploitation of water's unusual molecular properties. The cohesion of water molecules, held together by hydrogen bonds, where the partial positive charge on hydrogen atoms attracts the partial negative charge on oxygen atoms in adjacent molecules, creates substantial tensile strength in continuous water columns within narrow xylem vessels. Water molecules essentially pull on each other through these hydrogen bonds, transmitting tension generated by evaporation at leaves downward through the entire column to roots where water is absorbed from soil. This mechanism, called the cohesion-tension theory, was proposed by Irish plant physiologist Henry Dixon in 1894, but faced skepticism for decades because it seemed impossible that water could sustain the required tensions without the columns breaking. The adhesion of water to xylem walls, also mediated by hydrogen bonding between water molecules and hydroxyl groups on cellulose and other carbohydrates in cell walls, prevents the water column from pulling away from vessel walls, and helps support the column's weight through capillary action in the narrow tubes. In narrow vessels, typically 2,200 micrometers in diameter, depending on species, growing conditions, and location within the tree, these cohesive and adhesive forces overcome gravity and frictional resistance, allowing water to be pulled upward against its weight. Theoretical calculations based on the strength of hydrogen bonds and the molecular structure of water suggest water columns can sustain tensions of 30 megapascals or more before breaking, equivalent to supporting a water column 3,000 meters high or roughly 25 times taller than the tallest trees, so the system should work. In practice, xylem sap in actively transpiring trees is typically under tension of 0.52 megapascals during normal conditions, well below the breaking point, though tensions can reach 510 megapascals during extreme drought when soil water becomes scarce and difficult to extract. These tensions are genuine negative pressures, pressures below atmospheric pressure, meaning the water is literally under tension, being pulled from both ends. If you could somehow cut into a transpiring xylem vessel without disrupting the water column, which is essentially impossible because cutting introduces air, you would expect water to be pulled into the vessel toward the cut from both above and below as tension was relieved, rather than flowing out as it would from a vessel under positive pressure. However, this elegant system is vulnerable to catastrophic failure through cavitation or embolism, formation of air or water vapor bubbles in xylem that block water transport. When tension becomes too great, exceeding the tensile strength of water in a particular vessel, when xylem is damaged by freezing that forms ice crystals or by physical injury that breaks vessels, or when air is pulled through imperfect seals between vessels or through pit membranes, the porous partitions between adjacent vessels, the water column can break and air bubbles form. Once an embolism forms in a xylem vessel, that vessel ceases conducting water because the compressible air bubble breaks the continuous water column, and water must flow around it through adjacent vessels that remain functional. A single embolism in a single vessel isn't catastrophic because trees have thousands or millions of vessels in parallel providing redundancy. But extensive embolism affecting large proportions of vessels causes water stress, wilting, reduced photosynthesis as stomata close to prevent further water loss, and potentially death if the plant cannot supply water to leaves faster than transpiration losses and cannot repair embolized vessels. Trees have evolved numerous adaptations preventing or managing cavitation at both cellular and whole plant scales. Some species employ conservative water use strategies, closing stomata when water stress develops before tension reaches dangerous levels, detected through chemical signals sent from roots experiencing dry soil through the xylem itself. The hormone abscisic acid produced in roots travels through xylem to leaves where it triggers stomatal closure, reducing transpiration and the tension driving it. This prevents embolism, but also stops CO2 uptake and thus photosynthesis, creating the fundamental trade-off between obtaining carbon for growth and preventing desiccation. Other species tolerate significant embolism, losing 50% or more of xylem conductivity, without dying, relying on redundant xylem vessels and the capacity to grow new functional xylem in the next growing season to replace embolised vessels. Some trees can partially repair embolised vessels by dissolving bubbles under positive root pressure generated at night when transpiration is minimal. Though this mechanism is less effective in tall trees, where generating sufficient positive pressure to overcome gravitational potential and compress bubbles becomes difficult. Xylem anatomy represents an evolutionary trade-off between efficiency and safety that different species resolve differently depending on their environmental conditions and life history strategies. Wide vessels conduct water efficiently, moving large volumes with minimal friction because flow rate through a tube is proportional to the fourth power of radius. Doubling radius increases flow rate by 16 times, allowing rapid water transport to support high transpiration rates and fast growth. However, wide vessels are more vulnerable to cavitation for several reasons. Larger diameter provides more opportunity for nucleation sites where bubbles can form. Bubbles can spread more easily from one vessel to another through pit membranes if vessels are wide. And the longer vessel lengths associated with wide vessels mean more opportunity for damage that introduces air. Narrow vessels are safer from cavitation. Smaller diameter makes bubble formation and spread more difficult, and shorter vessel lengths limit damage propagation, but less efficient, requiring more vessels in parallel and more cross-sectional area devoted to transport to achieve equivalent conductivity. Species in drought-prone environments typically have narrow vessels prioritizing safety over efficiency because cavitation could kill the plant, making survival more important than maximizing growth. Desert trees and shrubs often have vessels under 50 micrometers in diameter, sometimes under 20 micrometers, sacrificing efficiency for the ability to function under extreme tension during droughts lasting months or years. Species in consistently moist environments have wider vessels prioritizing efficiency because cavitation risk is low and faster transport allows higher productivity and competitive advantage. Tropical rainforest trees commonly have vessels over 200 micrometers in diameter, some exceeding 300 micrometers, enabling high water transport rates to support large canopy areas and high transpiration. Temperate deciduous trees fall between these extremes, with vessel diameters typically 50 micrometers, balancing efficiency during the growing season against safety during droughts. The distribution of vessel sizes within a tree also varies. Smaller, safer vessels in roots where embolism would cut off water supply to the entire plant. Larger, more efficient vessels in stems where redundancy is greater, and intermediate sizes in branches and leaves where both efficiency and safety matter. A large oak tree can transpire 150-200 liters of water on a typical summer day, enough to fill a bathtub or provide daily water needs for a household. A mature rainforest tree might transpire 200 liters or more, with some giant tropical trees potentially transpiring 400 liters daily during peak periods when conditions drive maximum evaporation, hot temperatures, low humidity, bright sunlight, and windy conditions. However, actual rates vary enormously depending on tree size, leaf area, species physiology, and environmental conditions, and precise measurements are challenging because directly measuring transpiration from large trees is difficult. Methods include tracking sap flow through sensors inserted into trunks, measuring water uptake by roots in controlled conditions, or scaling up from leaf-level measurements multiplied by total leaf area, each with uncertainties and limitations. Nonetheless, the general magnitudes are well established. Large trees move hundreds of liters daily, and forests move millions of liters per hectare. The rate depends on multiple interacting factors that together determine the evaporative demand and the plant's response. Leaf area determines the surface available for evaporation. More leaves mean more stomata and more transpiration surface, with large trees having leaf areas of 200-600 square meters or more, many times greater than their ground footprint. A large tree might have 200,000 individual leaves, each with hundreds of stomata on their lower surfaces, and sometimes upper surfaces too, creating billions of tiny evaporative surfaces. Temperature affects evaporation rate through its influence on vapor pressure. Warmer air can hold more moisture, creating steeper vapor pressure gradients between the saturated airspaces inside leaves and the surrounding atmosphere that drives faster diffusion and evaporation. A 10 degrees C temperature increase roughly doubles the saturation vapor pressure, greatly increasing evaporative demand. Humidity affects the gradient directly. Dry air has low vapor pressure and creates steep gradients driving rapid evaporation, while humid air already saturated with moisture can accept little additional water vapor, reducing evaporation. Relative humidity below 30% drives much higher transpiration than humidity above 70%, all else equal. Sunlight drives photosynthesis, which requires open stomata for CO2 uptake, creating the necessary conditions for transpiration, and also heats leaves, which increases vapor pressure inside leaves and the gradient driving evaporation. Direct sunlight can heat leaves to 10 degrees C or more above air temperature, dramatically increasing internal vapor pressure and transpiration. Wind carries away moisture-saturated air near leaf surfaces, maintaining steep gradients that accelerate evaporation. In still air, transpired water vapor accumulates near leaves, increasing local humidity and reducing further evaporation, a self-limiting process. Wind removes this boundary layer, maintaining low humidity near surfaces and high evaporation rates. Hot, dry, sunny, windy conditions maximize transpiration, potentially exceeding 10 millimeters of water depth, equivalent per hour from forest canopies, rates that would drain soil moisture within days without continued water supply from roots or rainfall. The tree must continuously balance transpiration against water availability and uptake capacity. Transpiring too much without sufficient water supply from roots can cause progressive cavitation. Runaway water loss as damaged xylem reduces hydraulic conductance and forces remaining functional vessels under higher tension that causes more cavitation and ultimately desiccation and death. This balancing act occurs through stomatal regulation involving complex signaling pathways that integrate multiple environmental and internal signals. Guard cells surrounding each stoma, modified epidermal cells that form the stoma when they swell with water and close it when they shrink, respond to multiple signals transmitted through electrical, chemical, and hydraulic pathways. Light intensity and quality affect guard cells directly through photoreceptors that trigger opening in blue light, facilitating photosynthesis during daylight. Internal CO2 concentration influences stomatal aperture through direct effects on guard cells. When CO2 levels inside leaves drop because photosynthesis is consuming it rapidly, stomata open wider to increase CO2 uptake. When CO2 accumulates because photosynthesis is limited by light or other factors, stomata close partially to conserve water. Humidity sensed through changes in transpiration rate affects stomatal opening. Increasing transpiration typically triggers partial closure in a negative feedback that limits water loss, though the mechanism is complex and not fully understood. And chemical signals, including the hormone abscisic acid, ABA, produced in roots experiencing water stress, provides advance warning of drought. As soil dries and roots struggle to extract water, they synthesize ABA that travels through the xylem to shoots, where it triggers stomatal closure in leaves even before leaf water status has declined critically. This early warning system allows plants to anticipate water shortage rather than responding only after experiencing stress, providing time to adjust before damage occurs and reducing the total water loss during drought by closing stomata before full stress develops. However, closing stomata also stops CO2 uptake and thus photosynthesis, creating a fundamental dilemma during droughts that has no perfect solution. Remain open and risk desiccation through continued transpiration exceeding water supply, or close and risk starvation for lack of carbon fixation and sugar production needed for growth and even maintenance respiration. Different species have evolved different strategies along this continuum, representing different evolutionary solutions to the drought dilemma. Some species prioritize survival, closing stomata early at relatively mild water stress and entering dormancy or at least greatly reduced activity, waiting for drought to pass. These species may grow slowly and take decades to reach reproductive maturity, but they have high survival rates through droughts and predictably persist. Other species prioritize growth and reproduction, keeping stomata open longer and continuing photosynthesis and growth even as water stress develops, gambling that rains will return before mortality occurs. These species grow faster, reproduce younger, and may dominate rapidly after disturbances, but they suffer higher mortality during severe droughts, representing a high-risk, high-reward strategy. Neither strategy is inherently better. Success depends on environmental conditions, and particularly on drought frequency, intensity, and predictability. In environments where droughts are rare and brief, the aggressive strategy succeeds by maximizing growth and competitive ability. In environments where severe droughts occur regularly, the conservative strategy succeeds by ensuring survival through droughts even if growth is slow. In intermediate environments, both strategies may coexist, with conservative species dominating drier sites and aggressive species dominating moister sites, creating species turnover across environmental gradients and contributing to overall diversity. This diversity of strategies represents one of the mechanisms by which many species coexist in forests, partitioning the environment along drought tolerance gradients through different physiological strategies and trade-offs. Root systems are equally impressive as above-ground structures in their extent, architecture, and function, though hidden from view and thus less appreciated by people who notice trees primarily by their visible portions above ground. Tree roots extend far beyond canopies, often 2 to 3 times the crown's radius in lateral extent, and sometimes even further when resources are patchy and roots grow preferentially toward concentrated nutrients or water. A large tree might have root systems extending 20-30 meters from the trunk, exploring soil volumes of thousands of cubic meters, and competing with roots from neighboring trees throughout. Root architecture is as complex and diverse as canopy architecture, varying among species in the pattern of branch angles, branching frequency, vertical distribution in the soil profile, and responsiveness to local resource availability, and responding plastically to soil conditions encountered during growth. Roots grow preferentially toward higher nutrient or moisture concentrations, allowing efficient foraging for resources in heterogeneous soils. Most absorption occurs through fine roots less than 2 millimeters in diameter that maximize surface area for water and nutrient uptake, while minimizing the carbon cost of construction and maintenance. Fine roots consist mostly of living cells with high respiration rates and thus high maintenance costs, so producing excessive fine roots would waste carbon without proportional benefits. These fine roots are ephemeral, living only weeks to months before dying and being replaced by new fine roots growing from more permanent, thicker roots that serve structural and transport functions. This high turnover rate, commonly 30–80% of fine roots dying and being replaced each year, varying with species, climate, and soil conditions, represents a continuous investment of carbon and nutrients in maintaining an extensive, responsive, absorptive surface. Root turnover is a major component of below-ground carbon cycling, with dead root production often equaling or exceeding above-ground litter production. Studies using minor hysotrons and other techniques reveal that roots may contribute 40–60% of total annual plant carbon allocation in forests, a much larger fraction than earlier estimates based on harvesting roots suggested, because harvests miss the rapid turnover of fine roots that die between sampling events. Soil forms slowly over centuries through rock weathering and organic matter accumulation. The ground beneath our feet, the substrate supporting all terrestrial ecosystems, and the medium in which roots explore for water and nutrients, formed over immense timescales from solid parent rock through the combined action of physical, chemical, and biological processes working simultaneously but at vastly different rates and through fundamentally different mechanisms. Physical weathering breaks rock into smaller pieces without changing chemical composition through mechanisms including freeze-thaw cycles, where water-penetrating cracks expands approximately 9% upon freezing, exerting enormous pressure that pries rocks apart. Pressures can reach 200–300 megapascals, far exceeding the tensile strength of most rocks. Thermal expansion and contraction from daily and seasonal temperature fluctuations create stress along grain boundaries and microfractures, gradually weakening rock structure. In deserts where daily temperature ranges can exceed 30 degrees C, rocks sometimes split from thermal stress alone. Root growth exerts pressure as roots penetrate cracks and expand, wedging rocks apart with forces comparable to freeze-thaw pressures. Large roots can generate pressures exceeding 1 megapascal. And abrasion occurs where rocks collide during transport by water or wind, or where wind-blown particles scour rock surfaces, gradually wearing them down. Chemical weathering alters rock composition through reactions with water, atmospheric gases, and organic compounds, dissolving minerals and transforming them into new compounds with different properties. Water itself is a powerful weathering agent, particularly when it contains dissolved CO2 forming weak carbonic acid. Natural rainfall is slightly acidic with pH around 5.6 from equilibration with atmospheric CO2, providing a mild acid that slowly dissolves many minerals. The dissolution of calcium carbonate in limestone by carbonic acid, creating features like caves, sinkholes, and karst topography, illustrates chemical weathering's power. Given sufficient time, water can dissolve entire mountain ranges grain by grain, molecule by molecule. Organic acids produced by plant roots, Fungi, lichens, and bacteria accelerate dissolution by providing stronger acids than carbonic acid. Oxalic, citric, and other organic acids with pH as low as 3.4 can dissolve minerals much faster than natural rainfall, allowing organisms to extract nutrients locked in mineral structures. Some plants called hyperaccumulators are so effective at dissolving and absorbing specific minerals that they concentrate toxic metals like nickel or zinc to concentrations that would poison most plants, using the toxins for defense against herbivores, or in some cases excreting them to reduce competition from less tolerant plants. Hydrolysis reactions, where water molecules break chemical bonds in minerals, are particularly important for weathering silicate minerals that comprise most igneous and metamorphic rocks. When water reacts with feldspars, the most abundant mineral family in Earth's crust, composing about 60% of continental crust and 40% of oceanic crust, it breaks silicon-oxygen and aluminum-oxygen bonds, releasing cations like potassium, sodium, and calcium into solution and leaving behind clay minerals and silica. This transformation of hard, crystalline feldspars into soft, plastic clays dramatically changes rock properties, turning solid stone into material that can be worked by roots, burrowing animals, and even earthworms. Oxidation reactions where oxygen from the atmosphere combines with reduced minerals create another weathering pathway, particularly for iron-bearing minerals that oxidize to form iron oxides, rusting, giving weathered rocks their characteristic red, yellow, or brown colors. Freshly broken rock surfaces are gray or black because minerals are in reduced states, but exposure to atmosphere gradually oxidizes them, creating colorful weathering rinds visible in road cuts or quarries. Biological weathering involves organisms physically and chemically breaking down rock through secretions, penetration, and metabolic activities that actively attack minerals to extract nutrients. Lichens, composite organisms formed from fungi and algae or cyanobacteria living symbiotically, are pioneer organisms colonizing bare rock surfaces where few other organisms can survive. These remarkable organisms produce various organic acids, including oxalic acid that dissolve rock surfaces, creating tiny pockets of weathered material where moisture and organic matter accumulate. Studies show that lichen-covered rocks weather 2-3 times faster than bare rocks, and in some environments lichens are responsible for most weathering occurring. The organic acids etch minerals, preferentially dissolving specific elements the lichen needs: calcium, magnesium, potassium, leaving behind silica and aluminum that accumulate as small pockets of protosoil. Over decades to centuries, these tiny pockets coalesce and expand, gradually building a continuous soil layer that other organisms can colonize, initiating ecological succession from bare rock to complex ecosystems. Plant roots don't just passively exploit existing cracks and pores in soil, they actively penetrate rock, creating pathways where none existed before through combined mechanical and chemical action. Growing root tips secret organic acids, particularly mucilage, a gelatinous secretion that lubricates root tips and contains acids that soften and dissolve minerals ahead of the growing root, allowing it to push into small cracks or even create cracks in weakened rock. As roots penetrate, they follow paths of least resistance, existing fractures, grain boundaries, zones of weak minerals, but also create new pathways by dissolving cement between mineral grains and forcing aside particles through expansion. Once roots have grown into cracks, their expansion as they thicken increasing in diameter as the plant grows, exerts substantial pressure that fragments rock and widens cracks. When roots die and decay, they leave channels, macropores, that persist in soil, providing pathways that subsequent roots preferentially follow, gradually expanding the network of root channels and accelerating rock disintegration. These root channels also provide pathways for water infiltration and gas exchange, improving soil properties and supporting soil organisms that further accelerate weathering. Burrowing animals contribute to weathering by physically mixing soil, bringing subsurface material to the surface where weathering proceeds faster due to exposure to atmosphere, temperature fluctuations, and organic acids in surface organic matter. Earthworms, ants, termites, gophers, prairie dogs, and countless other organisms tunnel through soil, consuming soil particles and organic matter, grinding them in digestive systems, and depositing them in different locations. This bioturbation, mixing by biological activity, can completely turn over soil to considerable depths over timescales of decades to centuries. Darwin calculated that earthworms bring about 10 tons of soil to the surface per hectare per year in English pastures, meaning the entire plow layer would pass through earthworms approximately every 50 years, an extraordinary rate of processing that profoundly affects soil development. Modern estimates suggest Darwin's calculations were reasonable, with bioturbation rates in earthworm-rich soils often exceeding 515 tons per hectare per year, though rates vary greatly with earthworm abundance, which in turn depends on climate, vegetation, soil properties, and whether the soil has native earthworms or only invasive species introduced by human activities. Soil formation requires centuries to millennia depending on climate, parent material, topography, organisms, and time. The 5 soil-forming factors identified by soil scientist Hans Jenny in his seminal 1941 book Factors of Soil Formation, which revolutionized soil science by providing a quantitative framework for understanding why soils differ from place to place and how they develop over time. The often cited estimate that forming 1 inch of topsoil requires 501,000 years represents a very rough average that masks enormous variation across environmental conditions and parent materials. Volcanic ash in tropical climates can develop into productive soil within decades. Fine ash particles provide high surface area for weathering, tropical warmth and moisture accelerate all weathering processes, and rapid plant colonization adds organic matter quickly. Some volcanic ash soils in Japan, Hawaii, and Central America have developed a horizons over 50 centimeters thick in less than 1,000 years. At the other extreme, granite in cold, dry climates might take 10,000 years to produce a few centimeters of soil because weathering proceeds extremely slowly. Cold temperatures slow chemical reactions and limit biological activity. Low precipitation means limited water for weathering reactions and transport, and granite's coarse crystalline structure and low content of easily weatherable minerals makes it resistant to breakdown. Climate is particularly influential in determining weathering and soil formation rates through its control over temperature and moisture, the 2 master variables governing rates of chemical and biological processes. Hot, wet conditions accelerate chemical weathering reactions exponentially. A rule of thumb is that reaction rates roughly double for every 10 degrees C temperature increase, and abundant water provides the medium for reactions to occur and the means to transport weathering products away. preventing accumulation of products that would slow further reactions. Tropical regions with mean annual temperatures around 25-27 degrees C and annual rainfall exceeding 2,300 millimeters have extremely rapid weathering and soil formation, but they also have rapid decomposition and nutrient leaching, sometimes resulting in nutrient-poor soils despite rapid development. The intense weathering over millions of years leaches away all easily soluble elements, leaving behind only the most resistant minerals like quartz and kaolinite clay, plus oxides of iron and aluminum that give tropical soils their characteristic red and yellow colors. These highly weathered soils, called oxisols or laterites, can be meters deep, but are often infertile because weathering has removed most nutrients. Cold, dry conditions slow all processes: weathering, organic matter production and decomposition, nutrient cycling, resulting in slow soil formation, but potentially higher nutrient retention once soil forms because nutrients aren't leached away rapidly. Arctic and alpine soils develop slowly, often only a few centimeters per 1,000 years. But when they do form, they can be surprisingly fertile on a per-volume basis because nutrients accumulate rather than leaching, though the cold temperatures limit productivity regardless of nutrient availability. Permafrost, permanently frozen ground underlying much of the Arctic and some alpine regions, prevents drainage and soil development below the active layer that thaws seasonally, constraining rooting depth and creating unusual soil properties. Cryoturbation, mixing by freeze-thaw processes, disrupts soil horizons and creates distinctive patterns visible from the air, including stone circles, stripes, and polygons formed by repeated freezing, expansion, and sorting of soil particles. Parent material, the rock or sediment from which soil develops, exerts profound influence on soil properties through its mineral composition, particle size distribution, and resistance to weathering. These properties, established at soil formation's start, influence development trajectory and properties even after centuries of weathering. Granite, composed primarily of quartz, feldspars, and mica, weathers to sandy, acidic soils often low in plant-available nutrients because granite contains relatively few nutrient-bearing minerals. Most nutrients are locked in feldspars and micas that weather slowly. Granite soils typically have coarse texture, low water-holding capacity, and low base saturation, meaning most cation exchange sites are occupied by hydrogen and aluminum rather than nutrient cations like calcium, magnesium, and potassium. These properties limit productivity unless ameliorated through lime and fertilizer additions. Basalt, composed of ferromagnesian minerals rich in calcium, magnesium, and iron, weathers to clay, nutrient-rich soils supporting productive ecosystems. Basalt soils typically have fine texture, high water-holding capacity, and high base saturation, providing abundant nutrients. Volcanic soils in Hawaii, the Columbia Plateau, and other basaltic regions are renowned for fertility, supporting intensive agriculture and natural ecosystems of high productivity. Limestone, composed primarily of calcium carbonate, dissolves readily in acidic water through the reaction Ca-CO3 plus H2CO3 right arrow Ca2 plus 2HCO, producing fine-textured, neutral to alkaline soils often with high nutrient availability. Limestone dissolution doesn't produce much residual material. Most of the rock dissolves completely. So limestone soils are typically thin, composed of insoluble residues left behind or materials transported in by wind or water. However, the calcium released during dissolution provides a valuable plant nutrient and also maintains near-neutral pH by neutralizing acids, creating favorable conditions for most crops and many natural plant communities. Regions underlain by limestone like the Shenandoah Valley in Virginia or the karst regions of Slovenia and Croatia have characteristic landscapes with sinkholes, caves, disappearing streams, and springs where water resurges after flowing underground, along with thin but fertile soils supporting productive agriculture and deciduous forests. Glacial till, the unsorted mixture of particle sizes from clay to boulders deposited directly by glaciers without sorting by water, produces soils with highly variable properties depending on the rocks ground up by the glacier during its advance. Till derived from granite produces sandy, acidic soils similar to soils forming on granite bedrock. Till derived from limestone produces fine-textured, high pH soils. In regions covered by Pleistocene glaciers, till composition varies locally depending on bedrock geology, creating a mosaic of soil types that influences where forests, prairies, wetlands, and agriculture develop. Some of the world's most productive agricultural regions, including the North American Corn Belt, formed on glacial deposits that mixed diverse rock types, providing balanced mineral compositions supporting high productivity. Alluvium, sediment deposited by rivers, produces fertile soils because flooding deposits fine particles rich in organic matter and nutrients eroded from upstream, continuously renewing soil fertility. River valleys have supported agriculture for millennia because periodic flooding, while sometimes destructive, replenishes soil nutrients naturally, allowing sustained cultivation without fertilizer additions that would be required on uplands. The ancient civilizations of Mesopotamia, Egypt, India, and China all developed in river valleys where flooding maintained soil fertility, supporting dense populations impossible in non-alluvial regions. Modern flood control through dams and levees prevents flooding and thus prevents this natural fertilization, requiring fertilizer additions and sometimes leading to soil degradation as nutrients are removed in crops without replenishment. Topography affects soil formation through its influence on water movement, erosion rates, and microclimate variations that interact with climate to create local environmental gradients. On steep slopes, gravity constantly moves soil downhill through mass wasting, slow creep imperceptible on human timescales but substantial over centuries, plus occasional landslides and slumps that rapidly transfer large masses. This downslope movement removes soil faster than new soil forms from bedrock, weathering in many cases, resulting in thin soils or exposed bedrock on ridges and steep upper slopes. Soil eroded from slopes accumulates in valleys and lowlands, building thick, productive soils where water and nutrients concentrate, providing favorable conditions for plant growth and further soil development. This lateral redistribution of soil means that topographic position strongly influences soil depth and productivity. Ridge tops and steep slopes have thin, infertile soils despite being on the same parent material, and in the same climate as valley bottoms with deep, fertile soils. Slope aspect, the compass direction a slope faces, influences temperature and moisture through differential solar radiation receipt, affecting vegetation, and thus organic matter inputs that strongly influence soil organic matter content and nutrient cycling rates. The Northern Hemisphere, south-facing slopes receive more direct sunlight throughout the year, are warmer and drier, lose more soil moisture through evaporation, and support vegetation adapted to these warmer, drier conditions, often grasses or drought-tolerant shrubs and trees. North-facing slopes receive less direct sunlight, are cooler and moister, retain soil moisture better, and support vegetation adapted to these cooler, moister conditions, often dense forests with high productivity and organic matter inputs. These vegetation differences drive soil differences. South-facing slopes typically have lighter-colored soils with less organic matter, while north-facing slopes have darker soils with more organic matter. These differences are dramatic in mountain regions, where a few hundred meters of horizontal distance separating south and north slopes can create ecosystems as different as if separated by thousands of kilometers of latitude, with south slopes resembling ecosystems far to the south and north slopes resembling ecosystems far to the north. Time, the final soil-forming factor, is unique because unlike the other four factors, it always increases unidirectionally and cannot be manipulated experimentally, making it challenging to study directly except through chronosequences, sequences of soils of different ages formed on similar parent materials in similar climates, allowing time's effects to be inferred by comparing soils of known ages. Chronosequences reveal that soil development follows predictable patterns, though at rates varying with the other soil-forming factors. Young soils, recently exposed by glacial retreat, volcanic eruption, or other disturbances, show minimal horizon development, primarily A and C horizons with minimal differentiation. Parent material properties strongly influence young soils because insufficient time has elapsed for weathering to substantially alter composition. As time progresses, weathering gradually alters minerals, nutrients are leached from surface layers and accumulate at depth. Organic matter accumulates in surface layers from plant inputs, and distinct B horizons develop where translocated materials accumulate, creating increasingly differentiated profiles. In humid climates with active leaching, soil development eventually reaches stages where extensive weathering has removed all easily weathered minerals. Nutrients have been depleted through leaching. Clays have been destroyed by intense weathering, leaving primarily quartz and resistant oxides, and thick B horizons of accumulated iron and aluminum oxides have developed. These ancient, highly developed soils may be meters thick, but often have low fertility because nutrients exist primarily in surface organic matter rather than mineral reserves, and any disruption like clearing or erosion causes rapid fertility loss. The time required to reach this advanced weathering stage varies from thousands of years in tropical climates to hundreds of thousands or millions of years in temperate or cool climates. Some of the highly weathered oxisols in tropical Australia and South America have been developing since the Tertiary period, over 20-50 million years of continuous weathering on stable landscapes. Mature, undisturbed soils in temperate climates typically show distinct horizons visible in vertical profiles. Exposures of the soil from surface to bedrock, such as those seen in road cuts, foundation excavations, or soil pits, dug specifically to examine soil profiles. These horizons form as processes operating at different depths and rates differentiate the initially homogeneous parent material into layers with distinct colors, textures, structures, and chemical properties that reflect the history of processes operating over centuries to millennia. The O horizon at the surface consists of organic matter in various stages of decomposition. Fresh plant litter recognizable as leaves, twigs, bark, fruits, and seeds. Partially decomposed material where structures are fragmenting but still identifiable. And humus, the dark, stable organic matter so thoroughly decomposed that original sources are completely unidentifiable. And the material consists of complex organic molecules formed through partial decomposition and recombination of original plant, animal, and microbial tissues. Humus is chemically extraordinarily complex, consisting of thousands of different molecules, large, irregular polymers created through partial decomposition of plant lignins, proteins, and other compounds, condensation reactions that join smaller molecules into larger structures, and biological synthesis by soil organisms that transform original plant compounds into entirely new molecules. Despite decades of research, the exact composition of humus remains incompletely characterized because of this complexity. No two humus molecules are identical, and traditional chemical analysis methods that work for pure compounds struggle with such complex mixtures. Modern analytical techniques, including nuclear magnetic resonance spectroscopy and pyrolysis mass spectrometry, are gradually revealing humus chemistry, showing it contains aromatic rings from lignin, long carbon chains from lipids, nitrogen-containing groups from proteins, and various oxygen-containing functional groups including carboxyls, phenolics, and alcohols that give humus its reactivity and its crucial soil properties. Humus provides numerous benefits to soils, benefits so important that humus content is considered among the best single indicators of soil quality and capacity to support plant growth. It improves soil structure by binding mineral particles, particularly silt and clay, into aggregates, small clusters of particles held together by humus molecules that act as glue. These aggregates create pore spaces between them that allow air and water movement through soil, while the aggregates themselves hold water on their surfaces and within their interiors, creating a balance of aeration and water retention optimal for plant roots. Soils with good aggregate structure are friable, easily worked, drain well after rains but retain sufficient water for plants between rains, allow root penetration without mechanical resistance, and resist erosion because aggregates are more resistant to detachment by rainfall impact or runoff than individual particles. Soils lacking organic matter and aggregates become compacted when wet, hard when dry, drain poorly or dry out quickly depending on texture, restrict root growth, and erode easily. Humus holds nutrients in forms slowly available to plants through cation exchange. Humus molecules have many negatively charged sites that attract and hold positively charged nutrient cations like calcium, magnesium, potassium, and ammonium, preventing them from leaching away in drainage water, but allowing plants to access them by exchanging hydrogen ions from roots for the nutrient cations. Soils with high organic matter have cation exchange capacities 2-5 times higher than the same soils would have without organic matter, greatly increasing nutrient retention capacity. Humus also forms complexes with some micronutrients like iron and zinc, keeping them soluble and available despite pH conditions that would otherwise cause precipitation into unavailable forms. And humus slowly releases nutrients as it gradually decomposes. The turnover time of humus ranges from decades to centuries depending on climate and soil conditions. But even slow decomposition continuously releases nitrogen, phosphorus, sulfur, and other nutrients that plants absorb, providing a steady nutrient supply that reduces fertilizer requirements compared to soils low in organic matter. Humus retains water like a sponge, with high-quality humus capable of holding 4-6 times its dry weight in water compared to 0.51 times for clay and 0.102 times for sand. This increases the soil's total water-holding capacity and also increases the fraction of water available to plants. The water held loosely enough that plants can extract it through root pressure. Soils high in organic matter maintain productivity through droughts better than soils low in organic matter, even when both have similar texture. Because the organic matter holds additional water accessible to plants. This increased water-holding capacity also reduces irrigation requirements and increases the time between rains that plants can survive without stress, both economically valuable in agricultural contexts and ecologically important in natural systems where water availability limits productivity. Humus supports microbial communities that drive nutrient cycling, disease suppression, and countless other soil functions. Humus molecules provide carbon and energy sources for bacteria and fungi. The organisms also colonize humus surfaces using them as physical habitat. And humus modifies pH and other chemical properties, creating favorable conditions for microbial activity. Soils rich in organic matter have bacterial and fungal populations orders of magnitude larger than soils low in organic matter. Differences of 10-100 times are common. These larger microbial populations process organic matter faster, releasing nutrients more quickly, providing more plant growth-promoting substances like hormones and vitamins, and compete more effectively with pathogens, reducing disease incidence. The relationship is circular and self-reinforcing. Plants growing in organic matter-rich soils are healthier and produce more organic matter that maintains soil quality, while plants growing in degraded soils with little organic matter struggle and produce less organic matter, making degradation difficult to reverse without external inputs of compost, manure, cover crops, or other organic amendments. The horizon, or topsoil, mixes organic matter with mineral particles through biological activity, particularly earthworms, arthropods, and other soil fauna that consume surface litter and excrete it mixed with mineral particles from subsoil, creating the intimate association of organic and mineral matter characteristic of productive topsoils. This mixing occurs primarily through bioturbation, animal burrowing, root growth and decay, and freeze-thaw or wet-dry cycles that cause vertical mixing, rather than through any abiotic process. The horizon typically appears dark from humus content, color ranging from dark brown to nearly black depending on organic matter quantity and type, with more organic matter producing darker colors. This is the most biologically active layer, containing most plant roots which concentrate here because of favorable conditions, moderate bulk density allowing easy root penetration, high nutrient availability from decomposing organic matter and cation exchange, good aeration from stable aggregate structure, and high water availability from organic matter's water-holding capacity. Here is where most soil organisms live: bacteria, fungi, protozoa, nematodes, arthropods, and earthworms that drive nutrient cycling, organic matter decomposition, structure formation, and countless other processes collectively termed soil function. that determine the soil's capacity to support plant growth and provide ecosystem services. Bacterial populations in topsoil can exceed 1 billion cells per gram. Fungal hyphal lengths can exceed 100 meters per gram. And earthworm populations can reach 100–400 per square meter in favorable conditions, representing more biomass and metabolic activity than all above-ground animals in many ecosystems. The horizon is where organic matter is continuously incorporated into mineral soil through bioturbation. The mixing process creating an intimate association unique to soil that cannot be recreated simply by mixing organic matter with mineral particles in a container, because the natural process involves biological transformations that change both components. Loss of a horizon through erosion is catastrophic for soil productivity because these properties, high organic matter content, good structure, high nutrient availability, abundant organisms, take centuries to millennia to develop and cannot be restored quickly through any practical management, even with intensive inputs of organic amendments and careful management to rebuild structure and biology. Sites that have lost topsoil through erosion, overgrazing, or mining can show reduced productivity for decades to centuries, even with management inputs. And full restoration to pre-erosion conditions may be impossible if erosion exposed infertile subsoil or parent material. The value of topsoil is difficult to quantify economically, but is certainly immense. Estimates of the cost to replace topsoil functions with purchased inputs like fertilizers, irrigation, tillage, and amendments run into thousands of dollars per hectare annually, suggesting the natural capital represented by developed topsoil is worth tens to hundreds of thousands of dollars per hectare. Values that dwarf the market value of land in many regions and highlight the folly of allowing erosion to proceed unchecked. Sleep is approaching now if it hasn't found you already. The facts continue: forests transpiring water, roots exploring soil, mycorrhizae exchanging nutrients, decomposers transforming death into life, photosynthesis capturing light, stomata opening and closing in response to conditions that shift moment by moment. These processes operated before humans existed. They operate now as you rest, and they'll continue long after we're gone, regardless of whether anyone observes them or understands them or values them. Your existence depends on them absolutely. The oxygen you breathe, the water you drink, the food you eat, all provided by natural systems operating through physical, chemical, and biological processes that science reveals but did not invent. Understanding these processes changes perspective, revealing interconnections between yourself and trees transpiring on distant mountains, fungi threading through forest soils, bacteria fixing nitrogen in roots, rivers carrying nutrients from continents to seas. You're part of these systems, not separate from them, not above them, not exempt from the consequences of disrupting them. Rest well, knowing that nature continues its patient work through the night, building soil grain by grain, growing forests tree by tree, cycling elements atom by atom, maintaining the conditions that make life possible. See you next time.